So this is Catherine Lambrecht. This is the April meeting for the Illinois Mycological Association. If you are a lapsed member and haven't paid your dues, this is the time to take care of it, because the information on forays will be going out in a week or so, and they will only go to people who are up to date with their dues. So if you have a question about your status, you could Illinois Myco at MYCO, Illinois Myco at gmail.com. Ask them. Don't send the email to me, because all I will do is forward it to somebody. So don't ask for me. So our program tonight is Andrew Methven, who was Eastern Illinois University. And I remember at least Eileen Shetty, a longtime member, a long time ago. She and I drove down to his neck of the woods to join him for a foray. And I remember the smile on his face, because he was surprised that anybody showed up from Chicago, I'm sure. But, you know, Eileen and I, we were, you know, if we got him to say yes, we had to at least show that we were there. So I'm going to turn it over to Andrew Methven, but I want him to ask the question that I've been asking everybody now the last few months. What sparked your interest in mycology?
Wow. Well, thanks. First of all, thanks for having me. Yeah, you'd mentioned that, and it got me to thinking. And I guess where I'd start with this is that when I was growing up, I think I used to believe that there were a lot of, I don't know what to call them, coincidences. And I don't necessarily believe in coincidences like I once did. I guess what I'm trying to say is that I have a tendency to think, when I look back on life, that things have happened for a reason. This story is kind of one of those sort of examples of this. So I was an undergraduate student. I was a biochem major initially, but the thought of physical chemistry scared me. So I decided that maybe botany would be a better thing, and I'd just taken a botany class and did well. So, and I wasn't really sure what I was going to do with that. My last fall in school, I took a class called California Mushrooms, taught by a professor in plant biology, who now, when I look back on it, really didn't know all that much about mushrooms, but we spent a lot of time growing agaricus, the button mushrooms, in the greenhouses. And so we did that. We went out on some forays, and we even at one point went on a foray to Mendocino County. We met up with the classes from UC Davis and UC Berkeley and from San Francisco State. They had a fall foray in Mendocino. And I remember at that foray that there was this guy named Harry Tears at San Francisco State. And I remember in the evening, after we came back from being in the field, he would sit there and identify all the fungi. We went around the room and he would just go table to table and identify everything. And I thought, wow, that's amazing. That's where the story kind of ended for there. I graduated, didn't know what I wanted to do. At one time, I actually thought I was going to become a marine biologist. I was really interested in marine algae, and that didn't pan out. So I went to work in a plant nursery for about a year. And I started getting curious in, could somebody make a living in commercial mushroom growing? And I had some experience in that class, so I went ahead and applied for a few jobs, had a few interviews, none of which panned out. And the one that is the most pivotal was the last one, which I interviewed at a place called Far West Fungi, which was a operation that Rick Kerrigan worked for at the time. This is in the early 1980s. And some of you may know Rick because of his work on Agaricus and his monograph on the genus Agaricus. And so I interviewed with Rick, and Rick didn't hire me, which he and I have laughed about a lot since then. It's good when you can laugh about not getting hired for a job. but we got to talking, and he said, and he was saying he was working on his master's degree at San Francisco State with Harry Tiers. And I thought, that name sounds familiar from somewhere. So Rick said, well, you should contact Harry. And just to show you how long ago that was, those are the days where you actually like wrote letters to people and put them in the mail. So I wrote Harry a letter and told him I was interested in mycology, and he wrote me a very nice letter back and invited me to come visit him at San Francisco State. And the next thing I knew, I was enrolled in a master's degree program. And again, I don't think I really knew what I was planning on doing. My parents thought I was crazy because they said, what is he ever going to do with a degree in mycology? But fortunately, I got a teaching assistantship, and I taught general botany labs and absolutely loved it. And so somewhere during my master's degree program, I figured, well, I really like this. I don't want to teach at, high school or middle school. I'd really rather teach at the college level. So I knew I had to get a PhD. And then this is another one of those coincidence stories. So I really wanted to go to University of Washington in Seattle. I wanted to stay on the West Coast. And I applied and I got admitted, but they didn't have any funding for me. And I also applied at the University of Tennessee in Knoxville, and they offered me an assistantship. And so I packed up and moved to Tennessee, and here I am. So I look back on that, as I say, and realize that, a lot of those things happened, and I prefer to think that they happened for a reason. And the reasons were good. If we have time at the end, if you want to hear another story, I'll talk about my job at EIU, but let's talk about some fungi, because that's what you're here for. So I wanted to, I guess I should start this off by saying that I've collected a lot of spring fungi over the years in the Midwest, largely because I'm not particularly good at finding morels. I can take you to places where morels are. You can find them. I can be standing with them between my feet, and I have a hard time seeing them. So I know where to take you if you want to go. And you don't have to worry about me because I'm not going to find any of them. So anyway, what I ended up doing was collecting all the other stuff that I would find that was growing in the forest instead of the morels that everybody else wanted to find. So that's what I'm going to talk about. I'm going to talk about some of the fungi in the spring in the Midwest that I think have a tendency to oftentimes be overlooked. Some of them are going to be common. You're going to recognize them. Others of them, hopefully, will be a little more interesting for you. So hopefully there'll be something for everybody. All right, I'm going to break this up into two main groups. So we're going to talk about Basidiomycetes. So these are things like mushrooms that produce Basidia and Basidiospores. And then later on, we're going to talk about the Ascomycota, which produce acine ascospores. So those are things like cup fungi and morels and false morels. So we'll start with Basidiomycota. And I've got these in alphabetical order because it's easier for me. There's not necessarily any other rhyme or reason to it. So this is something that I would find fairly commonly growing in the grass. This is a Grossaby, or what I call a Grossaby dura. Some people refer to this as a Grossaby molesta. I don't think we've really sorted out the species complex yet. But the pale yellow pileus, you may be able to see a little bit of a partial veil around the edge of the cap, and a little bit of a partial veil on the stems. But then it's got a sort of a earth brown or clay brown colored spore deposit. Not an uncommon thing. And some of these things that I'll talk about don't just occur in the spring. So maybe spring and summer, sometimes spring and fall even. We'll talk about some examples of that as well. This exhibits one of these issues that a lot of people are frustrated with in mycology, and that's that the names keep changing. I thought things were complicated when I started studying fungi a number of years ago, but the names change almost faster than we can keep track of them nowadays. And this is one of those examples. So this is Candelomyces candolianus. So some of you might be familiar with this as Satharella candoliana, but it's got a new name. So honey brown pileus. Again, you can see the partial veil, the remnants on the edge of the pileus, but it has a dark spore deposit, sort of a purplish black to black color, and is usually on woody debris. So it can be on stumps, but oftentimes can be on broken down wood or chunks of wood, things like that. But typically grow in these dense clusters. One of the things that a lot of people find, because I see a lot of pictures of it, is Coprinellus mycaceus. So this is a yellow-brown Coprinellus with the striate margins, and you can't see it in the picture. Maybe if you use your imagination, you can. But there are all these salt-like granules all over the surface of the pileus, and this is one of the inky caps. And so these are just coming up out of the ground. The one on the left, you can sort of see how the edge of the pileus is starting to darken a little bit. So it's beginning to deliquesce. They auto-digest. And so not an uncommon thing to find in the forest. And again, so this looks like it's on soil, but it's really on fairly well-rotted wood. And the soil was just overlaying the wood, and then the fungus is coming up on the wood. Crepidotus crocophyllus. So most people, when they realize they have a crepidotus, they throw their hands up because they figure they're never going to be able to put a name on it. This one is relatively easy to identify if you get them when they're fairly young. So notice that the caps have sort of a peach coloration to them, as the gills do, and the brown scales on the cap. If they get older, the scales wash off and the color fades, and then it gets really complicated. But if you get them early enough, with the colors are still vivid, then they're relatively easy to identify. And you'll see this on several slides during the talk. So these are actually Michael Quo's pictures, because when I went back and looked through mine, all of mine were horrible. So Michael was kind enough to allow me to use some of his images. Gallerina marginata. So this is a spring and a fall fungus. I learned this as Gallerina autumnalis, and then the molecular data showed us that Gallerina autumnalis and Gallerina marginata are the same thing. And autumnalis, because it's autumnal, it comes up in the fall, and marginata because it's in the spring. That's the way it went initially. But as I say, it's all the same thing. But the brown pileus and the partial veil, sort of a rusty brown spore deposit, and again on wood. A lot of these things that I'm showing you are saprobes. So they're saprobic. They're litter decomposers, so they're on wood or they're on humus in the forest, and that's what they're decomposing. That's why I think some people don't go out and look for these things. For me, mycorrhizal fungi don't really seem to start till the middle of the summer, when it gets hot and humid. And so a lot of people aren't interested in these things because a lot of them you can't really eat, or there's nothing special about them. And so again, that's one of the reasons I guess I'm kind of fascinated by them, because I was always out looking for them. Gymnopus subsulphureus. So many people are familiar with Gymnopus dryophilus, and subsulphureus looks very similar to it, but the difference is really in the color of the cap. So the caps are yellow. They don't develop the brown coloration that we see in Gymnopus dryophilus. And you can't really see it in this image very well, but typically those what look like white rhizomorphs or threads at the base of the mushrooms are actually pinkish in color when they're fresh. And so, it's a pretty easy thing to recognize when you see it in the field. And this thing I commonly find just in the spring. Megacolibia rodmani. So this thing has to have an identity crisis because it's had so many names. So, you know, Colibia platyphila, Tricholomopsis platyphila, you know, the list goes on and on. But this is something that Ron Peterson and Karen Hughes described from North America. Turns out that we don't have the European species here. But again, it's on wood. You see the streaks on the pileus that has that sort of olive brown coloration. And then if you look at the lamellae, you can see that they're sinuate. They kind of curve in to the stipe. So the attachment of the lamellae is a pretty helpful diagnostic characteristic. Mycena gallericulata. So and this is one, and there's a debate that goes back and forth about whether people have Mycena gallericulata or whether they have Mycena inclinata. And most of the stuff that I looked at turned out to be gallericulata, the sort of pale brown colored cap. The mushroom on the left, you can see that, at least to me, the lamellae have sort of a pinkish coloration to them. And again, it's on wood. But microscopically is what you need to really separate the species, because inclinata looks a lot like this. But all these things, when I look at them under the microscope, they all come out to Mycena gallericulata. Panis concatus. So this is a pleurotoid fungus, but notice the violet to lavender coloration of the pileus and the lamellae and the stipe. The stipe is eccentric, which means it's off-centered. So you can see it sort of off to one side, kind of like a pleurotus is, what we see in pleurotus. That's where the pleurotoid name comes in. And this has gone under a number of different names, but Panis concatus is the best name for what we have here in North America. Pluteus petacetus. So most people are familiar with Pluteus cervinus. Cervinus has a brown colored cap. Pluteus petacetus typically is white, and notice the brown scales or fibrils on the disc of the pileus. And it's a little out of focus, but that mushroom on the left, you can see the pink coloration in the gills, and you can actually see that the gills are free. They're not attached. There's a band of tissue at the apex of the stipe. And then, yeah, here's Rhodotus palmatus. And Greg and I were talking about this earlier. Most of the field guides say this is rare. And Walt Sundberg, who was at Southern Illinois University in Carbondale, who Greg and Betty both know quite well. And I got to talking about this fungus one time, and we actually ended up writing a paper with Herb Moneson, who is at Bradley University, about Rhodotus palmatus in Illinois. And when we started looking for it, we found it all over the place. And as I was mentioning earlier, typically along stream banks and riverbeds where there were downed logs, and the logs would get inundated periodically with water. And when the water receded, then these things would begin to fruit, and they fruit on the underside of the logs. So you really have to get down and look for them. They're not on the top of the log typically, but on the underside of the log. And so we ended up finding it everywhere. Patrick mentioned earlier, it's not much of it in Chicago, but it's not an uncommon thing in the Midwest. And then Xeromphalina tenuipes. So most of us, when we think about Xeromphalina, these small white spored mushrooms, we sometimes overlook this one. And tenuipes is the largest, well, it produces the largest mushrooms within the genus. They're on wood, white spored. Some people might say this looks kind of like Flammulina volutipes, and indeed, the stems are fuzzy, kind of like Flammulina volutipes, but Flammulina volutipes has a viscid pileus, and this pileus is dry. And if you had a microscope, they have amyloid spores, and that's not what you see in Flammulina. So there are ways to separate them, but that's kind of what it reminds people of when they see it, and it's that size too. I mean, these are fairly large mushrooms. All right, well, I wanted to talk about a few other things besides mushrooms, so I thought I'd throw a few polypore and a few other things in too. So life was so much easier when I was an undergraduate and a graduate student, because everything was polyporous. And now there's not much that's left in polyporous anymore. It keeps being moved out. And so what I used to call polyporous squamosis, which is now serioporous squamosis, with the yellowish pilus with those brown scales, and if they were turned over, the large angular pores. This is on a standing tree. You can find these all year long. I mean, they're freshest in the spring, but they will last until the fall. I've seen them on the same tree in the fall when I've gone back there, and they've just kind of aged in place. Another thing that used to be a polyporous, so this used to be Polyporphys Arcularius, but now it's Lentinus arcularius. And again, angular pores that are fairly large, so you can actually see the individual pore mouths on those two specimens on the right. The brown scaly caps, and again, a lignicolous habitat. And then another one that I learned is Polyporphys alveolaris, that's now Neophilus alveolaris. So instead of being brown, it's kind of an orange colored fibrils on the surface of the caps. And one that's still in polyporous, maybe, at least for the time being, Polyporphys Umbilitis. So those are individual caps, each of which has its own stipe that are all united at the base into kind of a cluster, kind of like a cluster of flowers, if you want to think about it that way. Some people have tried to put this in another genus called Dendropolyporus, but it keeps going back and forth. So I don't know if we've really decided where it belongs at all at this point. The Basidiomycota also includes jelly fungi. So things like Auricularia. So this is now what we're calling Auricularia Americana. I learned this as Auricularia auricula. And there's still problems in this group, because there's a form of this that occurs on hardwood or deciduous trees, and there's a form of it that occurs on coniferous trees. But it's really difficult to separate the two. So some people call them, well, that's the deciduous form, or that's the coniferous form. But I think the name that we're using for this is Auricularia Americana. I put this in here just because I like the name more than anything else. So Ductifera pululawana. I can't even say that. But you know, both Auricularia and Ductifera have those gelatinous basidiocarps that they produce. And one of the things that I like about these, about the jelly fungi in general, is the fact that when it's wet and when they're rehydrated, they look like this. And when it's dry, they completely dry out and are oftentimes gone for all intents and purposes. You can't see that they're there anymore. And then it rains again and they rehydrate and they're right back to where they looked like before. And, you know, it's an amazing thing. It's an interesting strategy ecologically, because they have the potential to produce spores repetitively. So they rehydrate, they produce spores, they dry out. They rehydrate, they produce more spores, they dry out. They rehydrate, they produce even more spores. Most fungi don't do that, all right? Most fungi, it's like one time and they're done, and that's the end of it. And then here's another one. So this is Exidia glandulosa. So it forms these large flattened masses on down logs and branches. So you have individual Basidiomata that fuse together into these resupinate masses. And if you look at them with a hand lens, they have what look like what are referred to as glands on the surface. They're not really glands, but they're small dots or pimples that appear on the surface of them. And that's a really good field characteristic for that one. All right, now we'll shift over into something completely different. So we'll talk about some rust fungi. So this is another thing I got really interested in the spring when I couldn't find morels. I got interested in rust fungi, and many of you have probably seen this. Now in central Illinois, these things are common because the red cedar, Gymnopus virginiana, is really common on abandoned fields. And so it's one of the first woody plants that moves into abandoned fields, and you find these. And then in the spring, you find those growths on the branches that produce these gelatinous yellow orange structures that are called teleal horns, and those actually produce the teleospores. And this is an interesting fungus because it has an alternating life cycle. It alternates between the red cedar and malus, which is the genus for apples. So you find it on apples, but you also find it on crab apples. And so it alternates between the two hosts. It has to go back and forth between the two hosts. This is kind of somebody, if you're growing apples commercially, this is kind of a nightmare to have near you. So you probably go out and cut all the red cedars down for miles and miles, although I don't know if that's really a practical solution, but that's probably what you'd have to do. And I've always thought that these kind of reminded me of Christmas trees in April and May, when these little ball-like teleal masses are on the branches. And again, to show you that I get distracted by other things in the forest, on the left, you see spring beauty, Claytonia virginica. On the right, that's the actual stem of the Claytonia, and the little orange pustules you see are a rust. And so it's spring beauty rust, which is Puccinia maryi wilsonii. So when you're out looking for morels and you come across spring beauties, look at the stems and the leaves of the spring beauties and you'll find the rust. It's not on every plant, but you'll find it. It's not an uncommon thing when you really start to look for it. Most of you have probably seen this one. So you're thinking morels right now, and you're thinking Mayapples, because you've been told and heard that, oh, when you see Mayapples, it's time for morels, right? So if the Mayapples are knee high, then the morels must be around round two. But if you look on the upper surface, you see the whitish spots. But if you look at the lower surface, these orange masses that you see are the rust. And so the rust grows on Mayapple. It's Puccinia podophylla. Again, this has had several names over time. One of the things I should mention about both the spring beauty rust and the Mayapple rust is they're on just this host. So if you remember spring beauties, they come up, they don't last very long, and then they're gone. So what most likely happens, and the same thing's true with the Mayapple rust, what most likely happens is that the rust probably persists on the root systems of the spring beauties and the Mayapples. And then the following spring, when the Mayapple comes up again, the rust is already there because it was hanging out from the year before. They don't seem to necessarily kill them, but some people don't like the looks of them. It's actually a really fine ecological balance because the rust is obviously getting nutrients from the plant, which is photosynthesizing, but not to the extent that it actually kills it. So it wants to keep its host alive because it can continue to feed on the host while the host's alive. I mean, it's a brilliant strategy if you think about it from the fungus's point of view. And then here's one on Jack in the Pulpit. So Euromyces erytriphyllae. And so on the right, I had taken one of the leaves and bent it over onto a bench so that you could actually see the rust pustules. These are not as obvious. They don't really show up on the upper surface of the leaf, but if you turn the leaves over, you can see the pustules. And again, it's not on every one of them. So when you're out looking for morels, take a few minutes and look for some of these rusts that are on the that are on the spring ephemerals that we find. All right, so let's look then at some members of the Ascomycota. So I mentioned some of those. So these are things like Morchella. So there we have Morchella esculenta on the left, like Sarcosypha occidentalis in the middle, the scarlet cup. And so these differ because they produce acine ascospores. And so where the Basidiomycota produce basidia with external basidiospores, Ascomycota produce asci, these sac-like structures, and the ascospores are inside. And so there's a fundamental difference between the two groups when we think about the manner in which they produce their spores. So let's look at some examples. So Alluria aurantia, which we refer to as the orange peel fungus. So the orange coloration is a dead giveaway for this thing. Find it on humus and on soil. I didn't find it really commonly in Illinois, but I could find it almost every spring. I just didn't see a lot of it. So it does occur there. This is one which I found one time in central Illinois and went back to the same spot. Oh, I don't know, about 25 years in a row and never found it again. This is Dumontinia tuberosa. It used to be known as Sclerotinia tuberosa. And these little cups that are formed on a stalk, but if you notice at the bottom, the black thing that you see is a sclerotium. And so they produce these sclerotia underground in the fall, and then in the spring, the stipe develops, and then the soil actually came up right to the base of the cups. So when I found these, I actually had the common sense, which I don't always have, to carefully dig them up out of the ground. Something told me that there was more to it than just the cup. And so I managed to excavate the whole thing. And as I say, I found it one time and went back to exactly the same place year after year after year and never found it again. So Galeella rufa. So this one you probably have all seen too. And again, if you're out for morels, you'll find it, that kind of orange brown hymenium, the black fuzzy outer surface. And then if you cut them in half, the interior is gelatinized and on wood. And I always thought this was a spring fungus. And I remember the first time when I was in Illinois and I found it in the fall. And I thought, wait a minute, what is wrong here? And then I realized I stopped and thought about it for a while, and I thought, oh, all right, well, the temperature's about in the fall what it's like in the spring. Moisture, humidity is pretty similar. Okay, the fungus just has figured out a way to fruit twice a year as opposed to once a year. So you should find this one. This one continues to create some issues as to what the name of it should be. Some people have called this Helvella acetabulum. I think the better name for it is probably Helvella costifera. If you read the descriptions, Helvella acetabulum should be more of a yellow-brown color, and this is really a gray, and that's what costifera has. And so that's why I've used that particular name. And so again, this is one where it's oftentimes buried in the soil. So you need to be careful to dig them up so you get the entire stipe. Hymenocyphus fructigenus. So these small white stipitate cups. I find these common on hickory fruits, sometimes also on beech nuts, but for me, it's more common on hickory fruits. When I first started collecting these, I thought they were only a fall fungus, and which makes it hard because in the fall, when the leaves are falling, when this thing is fruiting, it's really difficult to find them because they're all covered in leaves. And then I remember one spring I was out walking in the woods and I looked down and saw these things and I was like, wait a minute. And there again was this thing that comes up twice a year in the fall and in the spring. Microstoma flocosum. So here you have these red stipitate apothecia, but notice the white hairs on the caps, on the cups, and then you can see the one where it's turned toward you, and you can see the fringe on the margin of the cups. I didn't find this thing commonly. It occurs in the Midwest, but I only found it a few times. I found a couple others I'll show you much more commonly than this one. I always got really excited when I found this because I didn't see it very often. Pachyella clipiata. So this you might think of as a Pisiza because it has kind of the appearance of a brown Pisiza. It's on decorticated wood, so that's wood that's lost its bark, and where the wood is really wet and beginning to decompose is where you'll find this thing growing. But the way in which I can typically recognize this is if you cut them in half, the interior is gelatinized, and Pisiza doesn't do that. So that's a really good field character to separate the two. And as I say, just slice them in half, and you can see this gelatinous band inside of the apothecia. So yeah, this one's had a lot of name changes too. So this is what I used to call Pisiza badio confusa, which I always really liked because I thought the name was really appropriate. There's a Pisiza badia, and then Dick Korf named Pisiza badio confusa, I guess because it was badly confusing when he compared it to badia. But then Don Feaster decided that no, it was probably the same thing as Pisiza phylogena, and now I think there's even another name for it. I think it's Phylocypha phylogena. I think is now the name for this. Not a Pisiza anymore. I mentioned some red ones, so Sarcocypha dudleyi, what a lot of us used to call Sarcocypha occidentalis. But dudleyi is probably the species that we're getting in most of the cases. So these bright red apothecia that don't have a stipe, and then Sarcocypha occidentalis, which has much smaller apothecia, but they're stipitate. So you can see this looks a lot like Microstoma flocosum, but there's no hairs, and so it's pretty easy to actually separate them. And then Urnula criterium, the devil's urn. So this is probably a species complex too. It grows typically again on the sides of fallen logs and branches. It produces this black mycelial mat. So if you look at the base of that image, you see the black there. That's actually kind of like a black felty mat. It's all mycelium that's growing in the surface, and those cups emerge from these. I've been seeing a lot of pictures on Facebook of this from Arkansas and South Carolina. So they're on their way north, I guess is what we'll say. Okay, well, I thought it wouldn't be fair if I didn't show you some pictures of some morels and some false morels. But I saved until the end. There, and as most of you know, there have been a lot of name changes in Morchella. And unfortunately, and I'm partly responsible for it, there's going to be some name changes in Gyromitra too. Because Andy Miller at the Natural History Survey and I have been working on these. Andy's been doing the sequencing and I've been doing the microscopy and trying to figure out what we're going to do with false morels. So stay tuned, I guess. So let's look at a few of these. So the first one, and this actually, most people put this in the Morchellaceae with Morchella, with the morels. And you're thinking to yourself, wait a minute, that doesn't look like a morel. And it doesn't. I agree with you. It looks like a Pisiza. It's cupulate. The one on the right, you can see that it's getting wrinkled. And typically what happens as these caps expand is they get wrinkled or veined. And that's a reasonably good field character to separate it from Pisizas, because Pisizas usually remain smooth, although I wouldn't bet on it. But the ascospores are smooth. That's a good characteristic if you've got a microscope to actually see them. So that's Dicyotis venosus and venosa. And again, I would find this in the spring when I went looking for morels. Again, not finding morels, but I'm finding Dicyotis. So then Gyromitra. So these are false morels. And I should give before I forget, I should give a plug for Alden Dirks. So Alden is a PhD student at the University of Michigan with Tim James. And Alden's looking for the presence of gyromitrin, which is the toxin that's produced by gyromitras. And he's looking more specifically for the gene that produces the toxin, because as some of you may know, in gyromitra, there's all this debate about which ones are toxic and which ones are not. And if you just look at the literature, you can't figure it out, because everybody says, oh, that I got sick off that, I got sick off that, or didn't get sick, and I've been eating those for years, or whatever it is. But what Alden's doing is he's doing sequencing of individual collections, but he's also doing genomic sequencing. So he's sequencing the entire genome for individual species, looking for a gene for the production of gyromitrin, trying to see if there's a way that you can correlate the compound with specific individual species, as opposed to what goes on right now. And as somebody who's been working with gyromitra, I can just tell you that the taxonomy is a mess. So, I mean, this is what I refer to as Gyromitra brunea. Oh, and if you want, I can send Catherine Alden's email address, because some of you may have seen it on Facebook. He made a request: if you find gyromitras, he wants you to send him the specimens, and because he wants to sequence them. And I should warn you, he's not going to give you your specimens back. He's going to put them in the herbarium at Michigan. But so keep some if you want it. But he will take more material. So there was just something on Facebook. I'll send I'll forward the email I have to Catherine, and it's got his contact information. So anyway, sorry, Gyromitra brunea. So I learned this as Gyromitra fastigiata. It has somewhat of a lobed or almost saddle-shaped appearance to it, and sort of a gradual wrinkling to it. It's not a really pronounced wrinkling. But Gyromitra fastigiata is a European species, and now we're calling that Gyromitra grandis. So fastigiata doesn't even occur in North America, and what we're finding is brunea. So you may find this one. The other one you may find is Gyromitra caroliniana. So this is much more brain-like, so it's not really lobed, but it's very strongly wrinkled and contorted. And these are, I don't know if you've ever found these are massive mushrooms. I mean, these can be, 15 centimeters tall and weigh a pound or more a piece. I mean, they're huge. And I've never eaten any of these. And I'm never going to. I remember running into a guy one time in the woods, and he had a bag with half a dozen of these in there. And we started talking, and I said, well, you realize those are potentially toxic? And he said, I've been eating these for 25 years, boy, and I never gotten sick. And I was like, okay. I'm not doing it, but what I was going to say about this one is I wanted you to notice this ascospore. This is from Michael's website. And notice this little bump on the end of the spore. You can't really see it very well with a light microscope. The spores are actually roughened. They have a reticulum, a network on the surface of them that's raised. But at the ends of the spores, there are these little appendages that stick out that are referred to as an apiculus. And both Gyromitra brunea and Gyromitra caroliniana typically have 1 to 5 apiculi at each end. I know, why 1 to 5? I have no idea, but it's the way it is. And you can actually use that to separate it from Gyromitra corphii. So look at the photo down here. Look at the apiculus. It's much more rounded. It's kind of knob-like or blunt, as opposed to the more pointy one. I know you're thinking, yeah, I've got a good imagination. that's a good way to separate them. Gyromitra for me is more box-like, so it's more square and more close to the ground. The epithecium is ground level. The stipe is oftentimes buried in the ground. And so those are the three most common gyrometras that we find in the Midwest. Although when you get up into Michigan, things begin to go a little bit crazy. So that's a whole different story for another time. I mentioned Morchella and its name changes. So again, we've all had to learn new names in Morchella. So Morchella angusticeps, the black morel with the black ridges, what a lot of us used to refer to as Morchella elata. Angusticeps seems to be the name we ought to be using. Morchella diminutiva, which have a more pointed epithecium, sort of this gray color, but as they get a little bit larger, they turn to yellow. So you can actually find varying stages of these all growing together where they're gray like this, and you can begin to see a little bit of yellow and tan starting to appear in these. And as they get a little older, if you let your morels sit there for a week and come back, they will have changed their color, but it's all the same thing. In the Southeast, and I don't remember if it gets up in the Midwest, it might, there's something called Morchella virginiana, and it's more egg-shaped and the apex is not pointed. So it's similar in color, but it's just a slightly different shape to it. And the molecular data supports them as being separate species. Morchella esculentoides. So this is what a lot of us used to call Morchella esculenta. And there's another species, good name, Morchella cryptica, the cryptic species, which looks just like this. You can't tell them apart morphologically. DNA sequences are different, but morphologically, it's all the same thing. And then Morchella punctipes, which I learned is Morchella semilibera. I like that name much better because to me, it explains what the mushroom's doing. So in most morels, you should probably realize that the margin of the epithecium is actually fused to the stipe. And you'll notice in these, it's not. The stipe is actually fused about halfway up on the inside of the epithecium. And it's got what we refer to as a free margin. And so that's really the best characteristic for it. I remember that. I hope I can tell this story. I remember the first time I found these mushrooms, and the person I was collecting with said, "You know what those are?" And I said, "Yeah, it's Morchella semilibera." And he says, "No, boy, those are peckerheads." And I was like, wow. So if you hear people talking about peckerheads, I think this is what they're talking about, at least from a mycological perspective. And then the last one I'll show you is Verpa conica. So Verpas are not as common. I find them, I would find them occasionally, and sometimes in weird places. I remember one spring I had a student who brought in some of these. She lived on a farm and she was walking through her dad's soybean field and found these things fruiting in the soybean field. It's like, really? But in the previous one, notice how the stipe's fused about halfway up in the epithecium, and notice that the stems are hollow. And if you look at Verpa, it's only attached at the apex and the stipe is stuffed. It's got these white cottony fibrils inside. And the reason I mention this is that a lot of people eat Morchella punctipes and people eat Verpa conica, but some people get sick when they eat Verpas. And so, people go out and they collect these and they find these and they just throw them all in the same bag. And then they get home and they eat them all and they wonder why they got sick. All right, so there you go. All right, and with that, I'd be happy to answer any questions if you have.
Well, I think as you've been talking, Greg, Patrick, Matthew have all been answering questions,
Oh, good.
which is great. By the way, you said you had you were going to comment about some of the work you're doing now or were doing recently.
Well, just with Gyromitra. So I guess what I would say, as I mentioned, Andy Miller at the Natural History Survey and I started working on this, he had a student that worked on Gyromitra for a while, and we published a paper. Then we did some work with some of the mycologists in Nova Scotia and published some things that we found on the Nova Scotian fungi, Nova Scotian species of Gyromitra. And then Andy and I have gotten interested, and again, I think it came about more because of what we had learned in Morchella, and how when we actually started doing sequencing of Morchella, what you found out was that there were a lot of different species that were undescribed in North America, and there were a lot of European species that do not occur here. And so if you think about names, most of our original names for fungi in North America came from Europe, because the Europeans came, they brought their names with them, they found these fungi, they said, oh, look, that looks like what I find back home. That's the name we put on it. Of course, we know that that's not the case anymore. We know when we start looking that we realize, no, this is not the same thing. And so that's kind of where Andy and I are on. So he and I have been working on, I've put together a list of all the taxa that have been named in Gyromitra, but that also includes things in Dicina and even a few Pizizas and some Helvelas that all kind of get lumped in there together. As I mentioned, I'm doing the morphology, Andy's doing the sequencing. We're trying to identify type specimens for as many of the species as we can. We've got a pretty good list that he has now sequenced. We probably have a half a dozen things we're still looking for. So some of those, there are types that exist. We just haven't gotten the types to sequence them yet, or I guess I should say to see if we can sequence them. And then there are some where they go back in the European literature into the late 1700s, early 1800s, and there may not even be a specimen. So we may have to designate a new type specimen to try and solidify what the concept of the species is. I mentioned the problem with Gyromitra fastigiata and how it's a European species and it doesn't occur in North America. Gyromitra gigas could be another one of those. There are things that have been called gigas from Michigan and Canada, but they're not the same as the European gigas. A new species. So yeah, I'll let the cat out of the bag. There's a new species coming down the road for what we used to refer to as gigas in North America. So, but again, that's what we're finding is that, a lot of these Gyromitra species that were described from Europe, they don't actually occur here. And anyway, we have a lot of work to do. So I just thought I retired, I guess, from being a professor, but I haven't retired from being a mycologist. So that continues. So
Well, life doesn't end at 65, as I understand.
No,
so
I've heard that too.
So we have some questions coming up. So this is from Mariah Rogers. Actually, she's got several, but we'll begin with the first. She goes, thank you for your talk and information. How does a mycologist approach establishing whether a rehydrating fungal fruiting body is producing more spores or isn't, and is just left over as usual? Can it be as straightforward as trying a spore print from a sample? Or should microscopy be done to see if the spores look, interact, fresh, viable, or they need to be proved to propagate the fungus in some controlled setting to actually establish they are viable?
how about all of the above? Um, well, yeah, I think a spore deposit would be one way to do it. And as I mentioned, I don't know if I said that, but with the jelly fungi, particularly if you think about it from an ecological perspective, it makes sense what they're doing. They're producing these rehydrated Basidiomycota when it's wet outside, and they're shedding their spores when it's wet. So the spores have a better opportunity to germinate and to grow. What's the point of shedding your spores if it's dry? All right, the spores are just going to lay there and they're going to desiccate, and that may be the end of it. All right, so I think the jelly fungi are onto something with this rehydration thing. I think you could get a spore deposit, but the other thing you can do if you have a microscope is you can actually look at take a piece of the tissue, and you got to stain it with either like something like floxine or Congo red, and because they're colorless, so you need some contrast. And you can actually find Basidia, and you can see the spores being produced on the Basidia. You can see various stages in development of the spores. So you could do it that way too. So yeah, I mean, I think there are ways to do it. Yeah. And isolating the spores would do it. But, we know polypores do the same thing. Polypores, some of the polypores that last, a long time, a few months, they seem to produce spores over a long period of time. Again, it's not just like a mushroom where it comes up, it's got to do with spores because it's going to disintegrate. Polypores last a while, they'll be around.
So Mariah again says, also, is our understanding that most fungi are one and done in their fruiting bodies based on that being more parsimonious claim and the fact that so many of them appear to rot away each year, or is it a claim based on this been systematically surveyed?
Well, I mean, I guess it depends on how you look at it. So, if you talk to morel hunters, they'll tell you they go back to the same place year after year. And the reason that they do is when they're picking the morels, they're not picking the mycelial mass that's underground. So it's kind of the analogy people use is picking fruit off a tree. The tree's still there after you pick the apples, right? So you pick the morels, you've left all that mycelial mass in the soil. And if the conditions are right, the environmental conditions are right, then they'll produce more morels the following year. So, I mean, I don't know whether that means it's a one and done thing, maybe one and done for that year. But I like to think of it as, those fungi are producing year after year after year after year. And that mycelium potentially is getting larger and larger and larger. So I don't think we have a good sense, at least I don't, of how long a mycelium lasts. We hear about the humongous fungus and how old it was and all that sort of stuff. But I think for most fungi, I don't know that we have a good sense of how long things will last. We can culture a lot of fungi, and they will last in culture on nutrient agar for a long time. You may have to transfer them periodically, but they'll continue to grow. Put them in the refrigerator. They're kind of like in cold storage. You bring them out and away they go.
Okay, well, Mariah once more. So recently I've noticed a lot of Trametes, versicolor, turkey tail are perfectly fresh as they were when I saw them last year. Strong with a fresh smell too. It made me wonder if that was one of the species that is actually resporulating, and that's why they would smell, keeping up the production of various protective chemicals. I assume that smell represents because these fruiting bodies are still making viable spores, or if I was off and they were just well preserved, fresh odor and all in the snow this winter. I had meant to look this up, but hadn't found the right way to search for the answer yet. In case you or Patrick or others here may happen to know, is T. versicolor known to rehydrate and send out viable spores multiple years from the same brackets?
Well, I guess from my perspective, not that I know of, but that's just me. I mean, I know they're persistent. I mean, they last a long time, and I've seen them in the spring where they look, I can't tell whether it's the ones from last fall or whether it's new ones. So I don't know. I don't know if Greg or Patrick has a better answer for that one.
I always assume that they're just looking good and leaving over, that they put up new ones for Termites, that they're not perennial, but I guess I've never marked one and followed it from year to year to confirm that.
Yeah.
Okay, Luna said, so in places like Illinois where temperatures and humidity levels might be similar in spring and in autumn, are there a lot of mushrooms that we think are spring mushrooms and vice versa that would show up in the fall?
Well, there are certainly some. I don't have an exact number, but I know that there are some that seem to have that twice a year sort of, appearance to them. And again, if you think about it, in the spring, like Galeella rufa, it produces cups in the spring and then they're gone. But remember, the mycelium's still in the ground, and all you need is the right conditions in the fall, and then they can come up again. And I don't know if that's more true of saprobic fungi that are on dead and decaying organic matter, as opposed to mycorrhizal fungi. Mycorrhizal fungi seem to have a narrower window of when they occur than saprobes do. And a lot of parasitic fungi seem to be pretty focused on one time of the year as well. So, but I've never actually counted how many of those. I guess that's something I need to do. I guess I need to sit down and go back through my data and start seeing how many species there are that actually sort of exhibit that kind of a phenomenon.
So Patrick and I have noticed that the dryad saddle pheasant bank, whatever you want to call it, pheasant back, we always thought it was a spring, and now we're seeing it spring and fall. And so it seems that its time is being expanded. In fact, I kind of see it all year long now. And so there seems to be some, we never had good data on it, but observationally, it looks like there's getting to be more things that have the biannual fruiting than used to.
So
Okay,
maybe what we need to do is see how we can tie this into global climate change. And
so what changes have been found due to these weather fluctuations that we are having in northern Illinois the last several years, which, Andy, you may not be aware, but at least in Lake County and McHenry County, we've had drought conditions. That might be reversing at the moment, but we've had drought.
well, yeah, I mean, I certainly think there's an ecological component of it. I mean, and I don't know that we know what this is going to do, but I think it certainly has the potential to change the range and the distributions of fungi. I mean, one of the examples that I can think of is Amanita tiersii, which was described from Texas, and then it was found in Arkansas, and then Walt Sundberg found it in southern Illinois, central Illinois, Indiana, Ohio. It's moving north, and I don't know if anybody's correlated environmental conditions with it, but it's a little bit different because it appears to be a saprobe in grass, as opposed to being mycorrhizal with a tree. But nonetheless, I mean, it seems to be moving northward, and so yeah, you wonder what's going to happen with some of these. Yeah, I don't know.
A question I'm sure that wasn't top of your mind, but what the heck? What was the top fungus of 2021, 2022, if there is such a thing?
Wow, I don't know. I mean, maybe we have to have everybody pick their top fungus. I'm trying to think what I saw here in North Carolina that I thought was interesting. Let's see.
Meanwhile, Heidi, you asked if there's any chance I could have a list of the mushrooms shared on this webinar. I think it's like, go back and watch it and then take notes. If you'd like, you could make that list available to everybody, but somebody has to do it, and time is everything. So if you're willing to do that, that would be lovely.
Well, I already have a list.
Oh, are you willing to put it together quick?
Oh, it's
not this moment, but later.
it's already in a computer file.
Oh, okay. You might get your
So-
dream, Heidi.
So
So anybody else? Do they have the, Greg or Patrick? Do you have the your big mushroom?
I did think of one fungus for 20, what was it, 2020, 2021? So I found it here in coastal North Carolina in the lawns. It's Agrossaby retigera, which was described, if I remember right, maybe from the Caribbean or maybe from Central America. I'm not sure. Or maybe it was South America. I think it's a Spegazzini name. And I think Singer transferred it to Agrossaby. But it's really cool. I showed you that that Agrossaby dura. It kind of looks like Agrossaby dura, but it has a reticulate pileus surface, kind of like Rhodotus, but it's yellow. So that was probably my favorite fungus for 2021. Just because I'd never seen it before.
Well, new is always fascinating, isn't it? Any more questions? I'm going through the chat now just to. Patrick, Greg, Matt, any comments you'd like to make?
I saw that Matt had said he was going to answer something about climate change and his favorite fungus. I'm sure it's a lichen, but that still counts.
No, I was just saying that Patrick was putting some answers in the chat, and I think Patrick had kind of said what you were saying about climate change causing some things to fruit a couple times a year, if I'm not mistaken. Is that right, Patrick?
Yeah, well, I keep track of the records of what we find in the Chicago area from the clubs and from our research trips, but there's been a long-term study in England by this guy, I forgot where, for 30, 40 years, he's been checking this one woods all year long and recording all the phenology of all the mushrooms. And he found some things recently have switched from only coming up in the fall to coming up in the spring and the fall. And one of those is the bluet, Clitospe nuda. And we find it here in the fall. And one of the people at the museum found it in the city in the spring. So that is now a spring and fall mushroom, for one example.
So I would just follow up with Andy's observation about most of the things that seem to have these changing phenology in spring and fall kind of biannual fruiting are mainly sap probes, especially if you're growing on wood, right? Because then you've got a steady resource, the wood's there. When it's wet enough and the temperature's right, there's nothing to prevent it from, it's as long as there's enough time for it to build up more resources, they can probably go ahead and sporulate, versus mycorrhizal things when we think of chanterelles and boletes and tricholomas and Licheria lactarius. Those are dependent on the tree to be, on the sugars that the tree is producing. So the tree physiologically triggers when it's shunting more carbohydrate to the fungus to trigger it to sporulate. So you probably won't see early spring bloom because the trees aren't producing the carbohydrates that the fungus needs. So I think we're going to see we see shifts in sporulating time, a time when the sporocarps come up, but they're probably not going to be bimodal like that, just because the trees probably aren't doing their thing yet.
By the way, James said, when I look for morels, I look for forested areas with lots of hardwood trees. When I've found them, however, it's been in parks and off the side of the road. Where are the best places to find morels?
Wherever they grow.
That was gonna be my answer, Andy. Exactly.
Well, I mean, I guess I can only speak for Central Illinois, but in Central Illinois, floodplains along the edges of the rivers and the streams were often good, particularly if there was ash growing there. Was usually a good place. Tulip poplars were good. I don't know if you get tulip poplars in Chicago or not. No, okay. I thought they were kind of limited to the eastern side of Illinois, but that's where tulip poplars are where I would go for black morels, because that's where I usually found them. But I had most success with ash in floodplains, but I've seen them in weird places. I mean, I was out at somebody's farm one day. We were doing something else, and we were walking down a road, and there was a road cut, and the morels were coming out of the hillside in a road cut. It was like, really? I mean, so yeah.
Yeah. So tulip poplars you can find if you just go over the edge and go into Indiana. So in the dunes area, there's tulip poplars, but they don't make it around the lake into northeast Illinois.
Oh, okay. Okay. Yeah, I knew they were in Indiana. I didn't know how far north they went.
Mariah comments, she says, I've heard soil temperature matters for morels. And somebody else comment, I was told to look for elm trees for morels.
Yeah, most of I've read articles about soil temperature and the correlation between when morels begin to fruit and soil temperature. I've never really spent a lot of time doing that, but I have noticed that when I've been out hunting for morels, you know, if I'm on a north-facing slope and I'm not finding morels, and I switch to the south-facing slope, oftentimes there are morels, and it's because of obviously the temperature of the soil because of the sunlight. But I've never really done anything with keeping track. I know people do that, but I've never really done that.
Yeah, and Hunter said she believes it's at 50 degrees where it matters. And then somebody says, I've only seen, oh no, I think, never mind. But I think, Greg, this is the moment to bring up your infamous morel story growing in your front yard.
Well, I don't want to make people cry. No.
You should
So
have been
some of
crying.
you have heard this story. So when I was growing up, our elms, so somebody said elms, and that's true. But as we know, there's not many elms around anymore. So we had this big elm in our front yard. It died. And for a number of years, these funny, spongy things popped up from where this old elm was. And the male person asked if they could take them out. I said, yeah, get rid of them. Take them, take them, take them. The year I realized they were morels, they never popped up again. So I never ate one of those morels. Isn't that a sad story?
It's amazing.
And the worst part is it turns out my dad collected morels when he was a boy, but he wasn't comfortable enough to risk it.
Amazing. Has anyone had luck growing Clitocype nuda?
I've heard a long time ago that you can, if you find it in the woods, you can collect a bunch of the leaf litter with the mycelium and bring it back to your yard and put it in a big leaf pile like oak leaves and stuff, and that might work. I don't know if anybody else has tried that.
Okay. Yeah, and I think I've heard apple and cherry both can be associated with morels. Just word of mouth learning though.
Yeah, old apple orchards have been told to be good, but you gotta be careful because some apple orchards, they use really nasty things, and that's in the soil. And so you just don't want to be eating all these pesticides. So you gotta be careful when you're doing that.
Yeah. Yeah, when I lived in Tennessee, and I used to go over to the Smokies to look for morels. I always looked in the old apple orchards along the edge of the park. But I agree with Greg. I would be concerned about what kinds of things have been used in the apple orchards and have been taken up by the fungi. Yeah,
In fact,
probably wasn't
Patrick,
the smartest thing I was doing. Yeah,
and Patrick said it might be arsenic in old orchards.
yeah.
I did it too, Andy, before somebody warned me.
Well, I was young and I was invincible back then, so you know.
Yeah, well, and then somebody's talking about the seeds of the apples containing arsenic, but a lot of seeds have arsenic in them, I believe. Somebody asked, what about lion's mane? Is that something we find in this region? Oh, we do.
I don't think springtime.
Yeah, I generally don't think about it as a spring fungus, although I found it in Savannah in January, so but that doesn't really count. So
Well, I think we're just about done. Andrew, Andy, I'm very glad you were able to drop in with us tonight.
it's been fun.
It was fun to see you. And thank you, Greg and Patrick and Matthew, for answering questions as they were flying by with, 140 devices connected. There's a lot of questions that just sort of erupted. So we'll probably be doing this more in the future. And if there's any, three participants raised their hand, but I don't know what it's about. But I think we're good. Okay. And this recording will be available tomorrow. And thanks again, everybody. We'll see you next month.
Bye, everybody.
Thanks, Andy.
Yep. Good to see everybody. Take care.
Oh, nice to see
Thank
you as
you.
well.